This video is going to be a refresher on how to use a compound microscope. We're going to be using the microscope in many of our labs to look at a variety of creatures. If you have a clear understanding of how to use the microscope in the lab, you're going to spend a lot less time fiddling with it and have a better viewing experience. I'm just going to grab it and bring it over. Notice how I'm handling it here. One hand supports the base, and I have my other hand on the arm. This is an expensive piece of equipment, and it's also quite heavy. So handling it carefully will ensure that you don't drop it or bang it against anything. I'm going to start by showing you all the different parts of the microscope, and then we'll actually look at a slide. Okay, I'm going to start up here with the eyepieces. Notice that there are two eyepieces. Okay, you can see that they are adjustable. This is to adjust the distance between your eyes. And notice that the one on this side has an adjusting ring right here. Okay, this is to adjust between the difference in focus in your eyes, and this is called a dioptric adjustment ring. Next, we're going to look down at the stage. So down here by the stage, you can see we have the nose piece, and this has the objective lenses on it. The lenses you're actually going to use to see your specimen. There's 4 different ones on here. There are different bunch of different powers, and you'll see that they're different sizes, and they're also color-coded. The lowest power one is either a 3 or a 4 or a 5, depending on which microscope you have. This one's a 5, and it has a red band. Some of the other ones have a brown band. The next one is the 10 power. It has a yellow band. 40 power has a blue band. And the 100 power has a white band. You can see that the length of this is quite a bit longer. This is an oil immersion lens, and we're not going to be using this one much for any of our labs because our creatures are much smaller or much bigger than this. We don't need to use this one. So now we look down at the stage itself. You can see on the stage itself we have a clip that holds the slide. You can see it has a little spring-loaded arm here. And the stage itself, where the slide is mounted. And to adjust the position of the slide, there's a little set of knobs underneath here. So the bottom one moves the clip back and forth, so side to side. And the top knob moves the actual stage back and forth. And this allows you to position the slide exactly where you want to look at it so that the image is right over top of the aperture here. So underneath the stages is a condenser. And so this takes the light from the bottom and focuses it on the slide. It is adjustable to change the amount of light going onto the slide. The other way to adjust the amount of light shining through is this knob down here. Some microscopes have a switch that you can turn the light on and off. This one does not, so it's just more or less a brightness knob. So as you turn it, the light gets brighter. So 2 ways to adjust the light on the slide. Okay, next we're going to look at focusing. I'm going to turn the microscope to the side here so you can see the focusing knobs. So there are 2 focusing knobs. One is a coarse and one is a fine adjustment. The large knob on the outside here is a coarse adjustment. Look at the stage and see how much it moves. Okay, when I use that knob, that moves the stage a lot. And the one on the outside is a fine adjustment, and you probably can't even see the stage moving. It only moves the stage just a tiny bit. We use the coarse focusing knob for the lowest power of the lens, and we use the fine focusing knob for all the rest of them. The microscope lenses are quite long, and if you use a coarse focusing knob with a long lens, you could probably crack the slide. And we don't want that. Okay, let's turn this back, and let's look at something. Before we plug the microscope in, it's a good idea to check to make sure that your light is turned off, if it has a switch, or if it just has the switch without a knob without a switch, turn the light all the way down. There's a slight chance that when you plug it in, a power surge might break the bulb in the microscope, and we don't want that to happen. So I've turned this down, and now I'm going to plug it in. And then we can have some light, and we are in business. Okay, I have a slide of some Daphnia here, and I'm going to put it on the stage. Now my stage is turned all the way down. Okay, and I have the lowest power lens in place. A trick that you'll notice when you move these lenses is, can you hear that? Actually makes a sound. So make sure that the lens always clicks into space, otherwise you're not going to be able to see anything. So lowest power lens, putting the microscope slide on the clip. Make sure that it's in the clip all the way. Try not to let this little spring-loaded arm snap back because there's a chance that that might break the corner off the slide, and we don't want that. And I'm going to turn the light on, and I'm going to move the slide into position. You want your specimen to be right in the center of that aperture. And so the light helps you to do that. Let's talk about light for a moment. Okay? You can have too much light or you can have too little light. Too much light makes it hard to see the details in your specimen. This image here shows a specimen with way too much light. It's hard on your eyes if you're going to be looking at a specimen for a long time, and you're not going to be able to see many of the details. This image shows an image that is too dark. Without enough light, you're obviously not going to be able to see anything. So let's actually look at our Daphnia. All right, we're going to start by adjusting the eyepieces for our eyes. So you want to make them just the right distance apart so you're going to get one image. They're one nice round image. Then we're going to adjust it for our eyes, our focus. So the focus is down. The stage is all the way down at the bottom. I'm going to use the coarse focus to move it up. If you have the stage somewhere in the middle, you may not know whether you need to go up or down to find your specimen in the microscope. So starting at the bottom, I'm going to cover my left eye, and I'm going to look through the right lens and focusing upwards through a whole bunch of stuff. And here comes my Daphnia, and I'm going to get it right and nice and tightly focused with my right eye. Okay, that's good. And now I'm going to close my right eye and use the dioptric ring to focus my left eye. So I'm not using the focusing ring anymore. I'm just adjusting this till the image is nice and tight. And when you open both eyes and look through, you're going to have a very nice crisp image. Okay? You can leave this setting here for as long as you're looking at anything. You don't have to readjust that. This is a shot of my Daphnia at 4 power. You can see that they're pretty big. I'd like to get a closer look, so I'm going to change my lenses again. So I'm going to go to the next highest lens, which is the 10. And notice that I'm rotating it to the 10. I'm not going to go the other way because the long lens, there's a danger that it could crack the slide again. So going to my 10, click it into place. And now you should only have to use the fine focus to get that precisely focused. We're not using the large knob anymore, just the small one. And there it's nice and focused. It's just a small adjustment. So this is a shot of my Daphnia at 10 power. You can see how much bigger it is. You can also see that as you increase magnification, the image, the part of the image you're looking at gets smaller. So you may have to adjust your slide a little bit to get the part that you want to look at. Okay, so I want to enlarge it still some more, so we're going to turn it up to 40. Okay, so that's the next one. See how much longer this lens is? Okay, we're getting pretty close here. So again, I'm just using the fine adjustment, and it doesn't take much to get that into focus. So now you see that this is an image of the Daphnia at 40 power, and it's a much smaller portion of the slide that we're looking at, so you may have to adjust the position. At this point, you may have to, and you may end up adjusting your light. Because the longer lenses, the light gets dimmer, so you may have to brighten your light a little bit. You can do that with the knob. Here, you can turn that all the way up. And if that's not enough, you can turn the little handle on the condenser here and allow more light in, just to brighten it up enough that you can see the details properly. Now, the depth of field is much shallower with a higher power lens, so you may find that you have to focus up and down to see other structures in your specimen. So the next lens, if I wanted to enlarge this still more, which I don't because it's already really, really big, I would use the 100 power lens. We're not going to use that here. It is an oil immersion lens, and it functions by putting a little drop of oil at the bottom of the lens, between the lens and the slide. So there's actually a little film of oil between the slide and the lens. But we probably don't have anything that's that small that we need to use that for. So now I'm done looking at my Daphnia. I'm going to reset my stage down to the bottom here. I'm going to reset my lens to the lowest power. Oh, where'd it go? Here it is. Okay, I'm going to take my slide off very carefully and turn the light down or off. And then I would unplug the cord, and I would wrap the cord around the base, put the cover back on, and put the microscope away in the cupboard. If you follow the procedures I've outlined in this video, you should have many hours of trouble-free creature viewing in the lab.